John has had a very long-standing research interest in diabetic and obesity-related dyslipidemia, with special interest to HDL function. And he's been interested in differences, sex differences in obesity-associated non-alcoholic fatty liver disease and cardiovascular disease, as well as understanding the effect of by which therapeutics impact on non-alcoholic fatty liver disease and cardiovascular disease. He is the present holder of 2 R01 grants, one looking at trying to understand the contribution of muscle and liver estrogen on signaling in fatty liver and disease and in dyslipidemia associated with obesity. The other grant is looking at trying to define CETP, cholesterol ester transfer protein, effects and sex differences in insulin-resistant states, and understanding how it impacts on lipoprotein metabolism. He also holds a DDA grant as a visiting professor, and hopefully when COVID-19 is over, he will join us at the Steno Diabetes Center in Aarhus. And it is on that behalf that I herewith hand over the microphone to John to give his today's talk.
God eftermiddag, tak skolen og Hanna. Jeg taler kun lidt dansk, så nu taler jeg på engelsk. We were supposed to have been with you guys this spring, as Soren said, and we literally had all of our bags packed and were ready to get on the plane in 2 weeks, and then COVID hit, so we were sad not to join you, but hopefully soon, and I hope you're all in good health. Yeah, so thanks for that introduction. Soren introduced the topics of my lab. The one I'll be talking about today is mechanisms for male-female differences. I'm just going to set my pointer. Mechanisms for male-female differences in metabolic and cardiovascular risk with disease. Soren mentioned we study a lipid transfer protein called CETP, which connects obesity-related changes with the dyslipidemia and atherosclerosis. And we're fortunate also to have a grant on COVID and type 2 diabetes. So this graph is a 20-year follow-up of the Framingham Heart Study, and it looks at incidence of coronary heart disease, really heart attack risk. And you can see in orange is the risk for men versus women, and you can see that women have about half the risk of having a heart attack in this 20-year follow-up period as men. And this sort of bluish green is people with type 2 diabetes. And you can see for men that the presence of type 2 diabetes about doubles coronary heart disease risk. For women, the presence of diabetes also markedly increases risk. But one thing that's very interesting is that in orange, you can see that women have marked protection. In green, there's no sex difference. One way to potentially interpret this data is that maybe something about diabetes takes away or gets rid of the protective effect of being female. And that's been an active research question of our group. When you look at data like this for the trainees in the audience, of course, you look at it with an aim of knowing what's known and what's presented. But the other important thing that you should try and take out of it is what's known, what's unknown, because those are really the opportunities for discovery. And in this graph, the first opportunity is we really don't know the pathways that confer this dramatic protection against coronary heart disease for women. The second thing is if diabetes takes away that protection, we certainly don't know how the mechanisms for that. This is just showing the dramatic impact of diabetes on coronary heart disease in a different way. Over a 20-year follow-up of the National Cholesterol Education Panel, you can see for women without diabetes, risk is very low. With diabetes, you can see the risk goes up markedly, and there's a really pronounced interaction of age with diabetes on heart disease risk. So some terms that are important for this talk. One is sex. Sex in here, in this case, refers to the biological and physiologic characteristics that define males and females. Gender is something we can choose for ourselves. It's the socially constructed roles and behaviors. A sexual dimorphism, there actually aren't a lot of these in medicine. A dimorphism is something where there's two options. And so, for instance, prostate cancer, ovarian cancer are examples. But almost everything else, whether we're talking about obesity, diabetes, heart disease, we really should be using the phrase sex differences. Now, many diseases have a sex difference. So rheumatoid arthritis, lupus, other autoimmune diseases have a strong female predominance. Autism, Parkinson's has a strong male predominance. This is from a nice article by Sabra Klein, where she shows, again, autoimmune disease is much more common in women, some non-reproductive cancers much more common in men. Interestingly, there are a number of infections that are more common in men versus women as well. And in fact, as her group has shown, there are sex differences in the function of almost all types of cells in the innate and adaptive immune systems. Even diseases that have a similar clinical incidence can have very different clinical courses. So here is showing survival after people have cardiomyopathy, which is much better for women than men. And so when we talk about sex differences clinically, this really represents the integration of the actions of sex hormones, but also genes expressed on the sex chromosomes, as well as lifestyle and environmental factors. So our group has mostly been interested in sex differences in obesity-associated risk. And as this group knows well, for any given body mass index, women have less cardiovascular disease. They have better lipids with lower triglycerides, higher HDL, and decreased liver fat. And much of that is due to the well-known different deposition of adipose tissue subcutaneously versus viscerally for women versus men. But our lab was interested when we started our lab many years ago in understanding what other tissues might contribute. And we really felt that this protection was against complications of obesity. This is again follow-up of the Framingham study looking at heart disease risk in 3 different weight categories for men. And you can see that men in the highest weight category have almost triple the risk. Women, here shown before menopause, as body weight goes up, also have an increase in risk, but it's much more modest. So what are the pathways that confer that protection? We started to look in liver because the liver is the organ that integrates glucose metabolism, lipid metabolism, and cholesterol metabolism. And at the time we were doing these studies, we hypothesized that there would be beneficial actions of estrogen signaling in the liver to promote fatty acid oxidation, that was known at the time in skeletal muscle, and also to promote the lipidation step of VLDL. Adding triglycerides onto ApoB100 to make VLDL is a major way that the liver protects itself from fat accumulation. And it was known that several egg-laying species had estrogen-like pathways that facilitated triglyceride secretion from the liver to promote egg development. And we thought, well, if that's evolutionarily conserved, these two biologies of fatty acid oxidation and getting fat out of the liver in the form of VLDL might protect the liver from fat accumulation with obesity. And so our objective was to define the relationships between estrogen signaling, insulin signaling with regard to glucose and lipids simultaneously. To do that, we used a mouse study where we made mice that have a hepatocyte deletion of the main estrogen receptor involved in metabolism called estrogen receptor alpha. We bred these mice to an albumin-Cre line to generate hepatic knockout mice, and the mice were compared to their littermate controls where ER alpha is floxed but present. This results in about a 90% reduction in ER alpha from liver, but no change in ER alpha in other tissues. Now, we did a number of surgeries where we could look at estrogen action where there's no estrogen at all, which is ovariectomy. We compared that to a sham operation, or we would give estrogen back systemically through a pellet, and that would give estrogen back to all tissues here except for the liver in the ER-alpha knockouts. An important primer that I'm sure you guys are aware of, when you're talking about the metabolic pathways in the liver, insulin is a potent signal to store energy. So insulin suppresses hepatic glucose production, and insulin promotes fatty acid synthesis and blocks VLDL secretion. That's normal insulin action. With obesity, there's this interesting physiology of selective insulin resistance where insulin fails to restrain gluconeogenesis. So this is on despite high levels of insulin. Even though the liver is insulin resistant with regard to glucose, lipogenic steps still remain intact. And so this selective insulin resistance is very important for obesity-associated heart disease because it gives rise to high blood sugars, but also to fatty liver and dyslipidemia. And so we wanted to be able to study both those glucose aspects and lipid aspects simultaneously in the same animal. And we actually adapted some tracer studies approaches that Soren and other labs around the world are very good at in humans. So for this study, we placed catheters in the carotid artery and jugular vein of the mouse. And then we can infuse tracers to look at baseline hepatic glucose production, VLDL production, and then we infuse insulin at roughly a postprandial dose, and we can look at insulin's ability to turn off both aspects in the liver. Now, insulin would normally lower the blood sugar, so to keep blood sugar normal, we infuse glucose at a variable level, and that glucose infusion rate is an index of insulin sensitivity. Here's our study design, which I just went through of the CLAMP study, but I'm also showing Western blots for protein of ER-alpha levels from liver, which you can see is reduced in liver ER-alpha knockout mice, but ER-alpha levels weren't changed in skeletal muscle or adipose. Now, the goal of the CLAMP is to keep blood sugars the same in the groups. This group actually, the study had many studies, including several controls not shown here, but you can see that the blood sugars are the same for sham, ovariectomy, ovariectomy with estrogen treatment, or ovariectomy and estrogen treatment on a background where there's no ER-alpha in the liver, again compared to the floxylitamates. I'm just going to show you the tracer data for hepatic glucose production on the top and VLDL production on the bottom. So compared to the baseline period before insulin was started, you see that insulin shuts off hepatic glucose production. These are the sham mice. Insulin also shuts off VLDL production, and this is normal insulin action in the liver. The overactomized model removes estrogens and creates a menopause-like model, creates insulin resistance with regard to glucose. So insulin does not shut off hepatic glucose production. Baseline VLDL production was higher in the overactomized mice, consistent with fatty liver in these animals. But even though they were insulin resistant with regard to shutting off hepatic glucose production, insulin still turned off VLDL production. So this is an example in the same animal of selective insulin resistance. Estrogen treatment reversed that selective insulin resistance. It restored insulin's ability to suppress hepatic glucose production, but interestingly, it blocked insulin's ability to restrain VLDL production. So even though insulin levels are high at the liver is still cranking out VLDL. And that would potentially be good for the liver because it helped the liver continue to secrete VLDL and avoid fat accumulation. Both of these effects required hepatocyte ER-alpha. In the ER-alpha knockout mouse that had ovariectomy and estrogen treatment, those mice basically looked like ovariectomized mice. We looked at molecular steps. To do that, we repeated cohorts and treated them with and without insulin. And here I'm showing from whole liver two important pathways. The first is insulin's ability to dephosphorylate ACC. So that's an early step in lipogenesis. So insulin dephosphorylates ACC in sham and overactomized mice. Estrogen treatment prevented insulin's ability to dephosphorylate ACC. So that would basically prevent lipogenesis from happening. The other important insulin-controlled step is ApoB. Insulin normally degrades ApoB, which you can see in these Western blots and in the bar graphs. Insulin potently degrades ApoB in sham and overactomized mice, and estrogen treatment blocked insulin's ability to degrade ApoB. So the physiology is supported by the molecular biology here, where overreactomy creates a selective insulin resistance, where insulin fails to restrain hepatic glucose production, but still is intact with regard to driving several steps of fatty liver. Estrogen treatment seemed to be the opposite of selective insulin resistance, and it restored insulin's ability to work down the glucose arm, but interestingly blocked triglyceride storage. At the time we were doing these studies, this neat paper came out from Joel Finkelstein. He took healthy humans and he made them hypogonadal with a GnRH superagonist drug. So the men were hypogonadal, and then he gave them testosterone replacement back in a dose response. And he did this with or without an aromatase inhibitor, because he wanted to ask which effects of testosterone require its aromatization to estrogens. So men have estrogens, although at a much lower level than women. And amazingly, he found that the aromatization of testosterone to estrogen was important for several of the canonical actions that we think of testosterone doing, namely body fat deposition and libido. So we had the male mice who were the counterparts of the study I just showed you. So we put male mice that were wild type flox littermates or hepatocyte knockout of ER alpha on high fat diet and performed a hyperinsulinemic euglycemic clamp. Here I'm showing you the glucose infusion rates in the clamp, and lower glucose infusion rate means insulin resistance. This insulin resistance was really at the liver. Insulin failed to suppress hepatic glucose production. Again, we repeated cohorts with and without insulin to look at signaling. In the knockout mice, we found insulin failed to stimulate AKT in the glucose pathway, and that's consistent with the liver insulin resistance we saw by tracers. Insulin still was able to dephosphorylate ACC. Again, they were insulin sensitive with regard to lipid steps, and this associated with increased liver triglycerides and diacylglycerols. In this graph, I'm just reminding us that women are protected against atherosclerosis. So we wondered whether these estrogen pathways in the liver might be protective against HDL function and atherosclerosis. A bit of a primer on HDL, as we call it the good cholesterol, but it's really not the cholesterol that confers HDL's protective functions. It's largely its proteins and signaling lipids, which are more than half of the mass of HDL. HDL has several important functions. The most important are reverse cholesterol transport, which is the ability to take cholesterol from tissues, bring it back to the liver for secretion as bile, and it's a potent anti-inflammatory particle too. Interestingly, drugs that raise HDL cholesterol levels don't have the expected coronary heart disease risk lowering. Our model, based on what we knew from our own studies in the literature, was that sex hormones would influence transcription pathways in the liver that would influence both the proteins made by the liver that end up in HDL particles and confer its function, and also proteins that help the liver take up cholesterol and convert it to bile acids. So in this study, Lynn, who's a faculty member in my group, used again male and female mice with liver knockout of ER alpha compared to their flox littermates and put them on a Western diet for here a long time. These are on a C57 Black 6 background. After that, she looked at early atherosclerosis and HDL function. She repeated a short cohort with just 5 days of Western diet feeding so she could study the mice without any differences in body weight or adiposity to look at reverse cholesterol transport. So I'm going to show you several aspects of those studies here. This study, Lynn isolated hepatocytes from the mice and looked at the ability of those hepatocytes from the groups to take up cholesterol from HDL or LDL. And you can see that there's a large male-female difference where female hepatocytes take up cholesterol from HDL much better than that of males, and this was modestly reduced by loss of ER alpha in females. There was a male-female difference in the ability of hepatocytes to take up cholesterol from LDL, but no impact of ER alpha loss. In this study, Lynn is looking at the ability of HDL particles isolated from the groups to take up cholesterol from labeled macrophages. And you can see that loss of ER alpha for both the males and the females reduced the ability of these HDL particles to accept cholesterol. So here we're knocking out ER alpha from liver, but it's changing the HDL particles and their ability to accept cholesterol. This is looking at the whole reverse cholesterol transport process put together. We inject macrophages that have been labeled ex vivo with radioactive cholesterol, inject that into the peritoneum of the mice. The cholesterol that's radioactive equilibrates with the mouse's cholesterol, and you can look at clearance over time. So females effectively clear that cholesterol. Without ER alpha in the livers, females do that less well. There was no impact of ER alpha on whole body reverse cholesterol transport in the males. Here we're looking at radioactive cholesterol uptake into both the liver and in the feces on the right. And again, there's a big male-female difference, and in females, loss of ER alpha prevents the ability of cholesterol to be taken up by the liver and secreted into the feces and gallbladder. So that shows that ER alpha is important for reverse cholesterol transport. Now, an important caveat of these studies is they're on a wild type background, and wild type mice are very resistant to getting atherosclerosis. And so even after a long duration of Western diet feeding, these mice have really very little atherosclerosis. So I'll just show the quantification here. You can see that on a wild type background, males do have more athero than females, but the loss of liver ER alpha increased atherosclerosis. And this is much lower levels of athero than you'd see like in an LDL receptor or ApoE null mouse. So in summary of the data I'll show in this short talk, we found that estrogen signaling through hepatocyte ER alpha promotes whole body and liver insulin sensitivity and protects against fatty liver. I didn't show you all of that data by increasing fatty acid oxidation and increasing VLDL output. We've shown that some of these effects require control of fatty acid flux to the liver. Some of these are transcriptional effects of ER alpha, and some of them require a downstream transcription factor called SHIP. We showed that ER alpha protects against atherosclerosis both by regulating genes that are made and put into HDL particles and by regulating genes that are important parts of how the liver takes up and handles cholesterol. Many of these aspects were also true in male mice with obesity. So one fun avenue that we're doing now is a collaboration with a colleague, Brian Finnan, who works at a branch of Novo in Indianapolis, Indiana, and he's helped engineer liver-targeted estrogen compounds so we can selectively give estrogens to the liver and avoid systemic effects of estrogens in both males and females. And we've done this now in mice and have a grant that we're trying to get funded to do it in non-human primates and hopefully one day in people. So I'll just sort of close by saying a few pointers about my time in research. One is sort of mind the gap, and what I mean by that is look for areas in the literature and in medicine where we don't know what's going on, because those gaps are really the opportunity for discovering. And just a few career tips. The most important, I think, is to find your passion. I've had a number of trainees in our group over the year, and I don't feel like it's pedigree of education that's the most important, but to me, it's really the drive. It's that sort of fire in the belly that's the biggest predictor of success. Don't be in a hurry with your research. Research takes time. A couple things: advocate for yourself when you go to conferences, really work on networking. I like this idea of called DIME, which is dilemma, idea, message, example. It's a concise way of talking about your research to people who don't know what you're doing. And similarly for your writing, there's this concept of the SOCO, which is the single overriding communication objective. It's really critical as scientists that we become clear writers. Let yourself go in depth when you're writing, especially. I like to turn off all of my media for the mornings that I'm writing so that you can really think creatively. And then, of course, get comfortable outside your comfort zone. So that's all I have. We've got a really great group. This is how we do our lab meetings these days on Zoom. And I acknowledged Lynn, who did most of the studies that I'll show to you. So I'll just sort of close with this quotation from Oscar Wilde. And
thank you very much, John, for this very clear and interesting presentation on sex differences in lipid metabolism and cardiovascular disease. And I'd like everyone from the audience to come up with questions for John. As of now, I don't see any. I may start off with a very simple question. John, all your studies describe mechanisms and effects of treatment with estrogen and estrogen ER alpha knockout in the resting condition of these animals. Have you any data looking at whether energy metabolism is solely prevalent or things that can be seen only under, for instance, physical exercise or after a meal? I mean, there may be differences in how substrates are oxidized and channeled to deposition and stored. I don't hear your voice.
yeah, absolutely. And that's a lovely thing that I know your group has done so nicely with tracers. And we've tried to model some of that in the mouse. And there actually really are, both known and in our group, sex differences in many steps of nutrient absorption, fuel deposition, particularly into adipose, the release of those fatty acids in response to a meal. So humans spend a lot of our time in a postprandial state, especially those with diabetes and insulin resistance, because they don't clear meal-related nutrients well. And so we've been starting to study mice after forced gavage meals and in the calorimeter where we can look at metabolic flexibility. And there are definitely roles of estrogens in several of those steps, particularly at skeletal muscle, which is shown by Andrea Hevner.
Okay, thank you. And I have a question here from Knud Yderstræde, and I'll just read your question. And Knud asks, do you know whether there is a difference in glycemic control among females, i.e. higher glycemic level, versus males that could to some extent explain the lack of differences in coronary heart disease risk among the two sexes?
So without diabetes, you know, women in general have better glucose handling. The question I think we really don't know that much about is in the presence of type 2 diabetes, what accounts for it's definitely an elevated relative risk to the non-diabetics. But in terms of the ability to control blood glucose when insulin levels are deficient with diabetes, I'm sorry, I don't know enough to answer that question.
Okay, so there's another question here from Eugenia Carvalho. And how early do these differences start? Do you have these been mapped in, oh, 1 second, another one came in over here. How early, I start from the beginning of that question again. How early do these differences start? And have these been mapped in relation to these models?
Well, it's a good question. In our mouse model, these have alpha knockdown, really at birth, the albumin Cree expresses at E18.5. We have inducible models that we could look at those, but definitely there's a lifelong effect in the mouse model. I think the more important question probably really is humans. There's a mother-child study that's done in Bristol in the UK where they've really looked at that, and there are really major sex differences, especially in triglyceride handling, particularly as kids sort of get above age 8 to 9. And then there are other changes again in later puberty years, and then they sort of settle into adult patterns. And so those puberty changes are actually very interesting. And one of the things that this group probably knows better than I do is that when kids get type 2 diabetes, which is unfortunately increasingly common these days, they seem to have worse metabolic outcomes for a given number of years of diabetes compared to an adult who gets type 2 diabetes. And some of that is thought to be an interaction with the sex hormones that are very high at those puberty years. But definitely it's very important, the programming of it, at least from late childhood.
Okay, thank you. There's another question here from Nicholas Huron. And it says, hi, John. In the examples where you saw VLDL output was higher or increased, was plasma triglyceride concentration also higher or increased? And if not, why do you think this was mechanistically?
Yeah. Thanks, Nicholas. It's a really perceptive question. It does show you something that you can learn with tracers that sometimes you can't find just sacking mice and looking at triglycerides. So the triglyceride levels in the blood were a little bit higher in the OVX mice, and they're reduced with estrogen treatment, but not as markedly as the tracer data shows. The thing with the mouse is that the mouse's metabolic rate is 10 times higher than humans. So they're really good at clearing triglyceride-rich lipoproteins. Certainly in humans, particularly insulin-resistant women that you treat with estrogens, they often see a really profound increase in plasma triglycerides, and that's been known for many years. But you run into problems with the mouse because their metabolic rate is so quick, and so it does show an advantage of things that you can learn by using tracers. For people who study the mouse, and another sort of tip that I'd add, a lot of our results, we wouldn't have, particularly with regard to molecular pathways, we wouldn't have actually seen those results if we hadn't taken cohorts where we had them animals with and without insulin. If you only look at endpoint without insulin or endpoint with insulin, we really wouldn't have seen a lot of the changes that we see. You really have to look at the delta of the pathway, no insulin to insulin. That's what really led us to see, for instance, that the AKT pathway was regulated differently than signaling to ACC or SREBPs, which I didn't show, for instance. So that's just sort of a tip that we've learned the hard way over the years.
Okay, there's another question here from Jenny Liebeck from Aarhus, and she writes, thank you for a nice talk. Do you know how well the mouse model of sex differences in metabolism in relation to obesity corresponds to the sex differences seen in humans? I guess that the apple pear shape is not as pronounced in mice.
Right, the apple pear shape is not as pronounced. It's really hard to show those visceral fat depots in the mouse because you have to dissect them so carefully. There is a little bit of fuel deposition difference in the mouse. The other really major area where the mouse in some ways fails to recapitulate the human disease, well, there's a couple, I guess, is with regard to risk of atherosclerosis. So if you have mouse, a wild type C57 Black6J mouse, the males do get more athero than the females, and that's like humans do too. But most groups that study atherosclerosis study the LDL receptor null or apoenol background. And very interestingly, the sex difference is reversed there. So the females get more athero. And it's a very, very important distinction. We actually have an aim in one of our grants to understand, to try and understand rather why that sex difference gets reversed in LDL receptor null mice. The other really important point of the mouse is that it naturally lacks this lipid protein CETP. So normal mice have almost all of their cholesterol and HDL, whereas humans carry cholesterol and VLDL and LDL as well. So that's why we started using these CETP transgenic mice, because they have a more human-like lipid profile. So the mouse has a lot of disadvantages with regard to glucose homeostasis, fatty acid kinetics, tissue triglyceride storage. Those pathways are similar to humans, but just the rate, the metabolic rate is so much faster.
Okay, I don't see any more questions coming up in the chat at this point.
No, I do not have further questions either, but then I know from the other webinars that sometimes it takes a while to write a question. And meanwhile, I will open a small poll that I hope you will all take your time to fill in.
I'm certainly available by email. I'm just john.stafford@vanderbilt.edu for any further questions, and I'd be happy to have an ongoing conversation, hopefully in Denmark soon.
I might want to follow up on one of your answers to one of the questions, if possible.
Please.
Just to sort of follow up on the production rate of VLDL and the concentrations in plasma. Do you have any sort of feeling or correlation analysis or feeling for whether there's a correlation or a coordinated peripheral clearance that is higher in those mice with higher VLDL secretion rates that sort of could account for the lower triglyceride concentrations? Because in all our human studies, more or less, we see that whenever VLDL triglyceride secretion rate is increased, then the plasma triglycerides go up as well. And we don't really have any models where peripheral clearance goes up also. So I just wondered in your estrogen models that have any indications of whether muscle or fat tissue could take up triglycerides faster?
Yeah, I mean, I think that's the mouse-human difference I was alluding to. And certainly with obesity in general, we haven't simultaneously looked at triglyceride clearance into adipose, just because we sort of get limited by tissue sometimes with the mouse. But obesity decreases triglyceride clearance. In fact, there are a whole bunch of drugs that are coming out in some major New England Journal papers over the last couple of years, targeting ANGPTL3s, ApoC3, and so forth that are aimed to promote triglyceride clearance. Because Soren's exactly right. You have this sort of double problem in humans with obesity, where liver VLDL output is increased largely because of liver fat accumulation, increased fatty acid delivery to the liver, but clearance is down because of expression, for instance, of ApoC3 and other pathways that inhibit triglyceride clearance by lipoprotein lipase. So that's something that's a little bit harder to study in the mouse, but we would expect triglyceride clearance, definitely like you've shown in the humans, to be reduced in obese states.
Søren, do you have any further questions in your chat?
I don't have any further.
No,
I have polling in the poll.
Yes, neither do I. So we've already exceeded the 30 minutes with almost 15 minutes. So if there is no further questions or comments to John Stafford, I think we might wrap up. Thank you to all of you for attending the webinar and for your questions and We really appreciate that. And thank you to John Stafford, of course, and to Søren Nielsen for helping us introducing John and managing the questions. We will end the webinar. Yes. Bye-bye. And vi ses.